It's really a huge honor to be named as one of the recipients of the BBVA Foundation Frontiers of Knowledge Award. I feel truly humbled to be chosen by the selection panel from among so many eligible candidates. Since my school days, quite a long time ago, I have been fascinated by DNA, and it was therefore somewhat disappointing to be told at university that all the key experiments had already been done and it was just a matter of filling in the relatively minor details. Fortunately, this assessment turned out to be incorrect, and as a result, my colleagues and I have been able to study the functional significance of DNA methylation, which involves the selective addition of a chemical mark onto the genome. Back in the 1970s, we knew that this mark existed, but nothing was known about where it was placed or what it meant. And a long time ago, I used DNA cutting enzymes to generate the first DNA methylation maps. And this showed, fortunately, that methylation is added in a distinctive pattern. Most strikingly, methylation-free islands are apparent at the regulatory regions, the control regions of most human genes. So the question is, what does this pattern mean? And we now know that there are sets of proteins that read non-methylated sites at these islands and help to switch genes on. And there's another class of proteins that read DNA sites that are methylated and cause gene expression to be switched off. And in this way, DNA methylation patterns establish a so-called epigenetic landscape across the genome that determines the way genes work. But one important reader of DNA methylation is the protein MECP2, which we discovered in 1992. And MECP2 seeks out methylated sites and it helps to silence genes. In 1999, it emerged from research in the United States that mutations in the gene encoding this particular protein cause a profound autism spectrum disorder called Rett syndrome. And this unanticipated connection with disease gave added impetus and urgency to our work. By using animals that accurately model the human disorder, we could ask if Rett syndrome is reversible. This is possible because nerve cells in Rett patients are alive, albeit abnormal. It's not a neurodegenerative disorder. And to our surprise and delight, my colleagues, some of whom are here, took a leading part in this. We found that severe Rett syndrome symptoms are reversible if we restored the missing MECP2 gene, meaning that Rett syndrome, a neurological disorder, is in principle curable. So what we're now trying to do is use this information to find an effective therapy, and our dream remains that Rett syndrome will become the first curable autism spectrum disorder. Thank you very much.